I'm really excited to be here today. My name is Fradkin, and I'm going to be presenting Concerto, a graph neural network approach for molecular carcinogenicity prediction. And I'm a PhD student at University of Toronto and the Vector Institute in Bo Wang and Brendan Fry's labs. And this work was done by also my wonderful, talented co-authors, Adamo Young and Lazar Tanakovic, and co-supervised by Brendan Fry, Leo J. Lee, and Bo Wang. So first, I'm going to introduce carcinogenicity as a concept. So you can sort of think about carcinogenic compound discovery in two main modes, the really high effect size mode or high exposure. So a compound can be, for example, really high effect size, and an example of that is birthworts, which contains aristolochic acid. And in the early 2000s, it was used as a weight loss treatment, and then we sort of found out that this thing is extremely carcinogenic. Or you can think about the high exposure mode, where here on the right you have an English chimney sweeper, and in 1775, Sir Percival Pott identified this population of people to have really high rates of squamous cell carcinoma caused by benzopyrene, which we found out only later. So we can ask, what about compounds with intermediate carcinogenicity and exposure, which epidemiological studies might not be able to pick up? So an effective approach would increase the percentage of positives identified. It would be low cost and high throughput. So in our work, we propose an in silico compound selection approach for downstream experiments. So speaking of experiments, there are two sort of main ways of thinking about carcinogenic experiments. So on the left here are carcinogenicity experiments, where you have some sort of animal, usually a mouse, and you introduce a compound to it, and then you measure the tumor count in your case group versus control group. And the upside is that these kind of experiments measure genotoxic and non-genotoxic effects, where genotoxic refers to a compound's introducing carcinogenicity through DNA mutational processes. And an example of a non-genotoxic compound is thought to be, for example, alcohol. On the downside is that these experiments tend to be pretty expensive and slow, so there are few of them publicly available. Whereas on the right, you have mutagenic experiments, where you have a cell culture and they measure only genotoxic effects, where you have a cell population and your readout is cell growth. So you introduce a compound and you observe if there's more cell growth, then there's probably genotoxic effects. So on the plus side, these experiments are relatively cheap and abundant. They have lower rates of replication. So conceptually, carcinogenicity prediction can be thought of as graph level regression problems. And there is a few challenges associated with it. So the first one is that this problem is extremely data constrained, and there's only about 1,000 samples available. So a carcinogenicity, a molecule and a carcinogenicity experiment pair. And the other issue is that carcinogenicity is really a continuous phenotype, because it's a function of dosage. It really depends on how much compound you're introduced to. And finally, actually, it's a very complex phenotype. There are multiple molecular mechanisms, and we can't really tackle that in this work right now, because you really need more fine-grained data for this. But this is an important thing to keep in mind, I guess, for future directions. So I'm going to talk about our contributions. So on the data side, we reinterpret carcinogenicity prediction as a regression level problem. On the modeling side, we pre-train on related lower quality mutagenicity experiments that I mentioned. We conduct transfer learning from a large self-supervised model, and we're the first to our knowledge to apply GNNs, graph neural networks, to carcinogenicity prediction. And for interpreting this model, we generate counterfactual molecules for interpretation. So we can think about representing a molecule in a variety of ways. We can think about like a natural graph representations, where the atoms are nodes and edges are bonds. And there's also a variety of string representations for molecules, such as smiles or selfies. And one of the sort of difficulties in this representation is that there's a many-to-one mapping, meaning a single molecule can have many kinds of strings. And if you want to design a model, you want your model to be really invariant to the identity of the strength if they both map to the same molecule. And then finally, there are binary vector representations referred to as fingerprints, and these bitwise vectors indicate molecular subgroup membership. So whether your molecule contains particular subgroups. And then, so in our work, we utilize 3 data sets, 2 carcinogenicity and 1 mutagenicity. And as I mentioned, mutagenicity experiments are more abundant, so our mutagenicity data set is bigger. And only one of our carcinogenicity data sets has continuous measurement, which is TD50. It's a measure of tumor count normalized to dosage as a proportion of animal body weight. So this is the high-level architecture of our model. So it has 3 main parts. We make use of Grover, which is a GNN transformer. Then there, in green, there is this multi-layer perceptron fingerprint predictor. And finally, we train this representation using multi-round mutagenicity pre-training. So the first part in blue here, it's a 100 million parameter model trained on 10 million molecule data set by Yurong et al. From 2020. At a high level, it's a graph neural net whose output is fed into a number of transformer layers. So it learns a local neighborhood and a global molecular representation. And it's a model trained with self-supervised learning on molecular graphs. And then the second component is an MLP fingerprint. So it's a concatenation of the molecular fingerprint vector that I mentioned earlier with the Grover representation, which is then passed to an MLP. And it contains individual carcinogenicity and mutagenicity prediction heads. And the way it's trained is we pre-train our model on these lower quality mutagenicity data, which is a classification task. And then we train the model on carcinogenicity data, which is a regression. And then we find actually if you repeat this process multiple times, the model is able to learn to have better regression accuracy for carcinogenicity data. So the full model is trained end-to-end using backpropagation. And another common convention is that these Grover weights are frozen, which is common in large NLP tasks. So now I'm going to talk about some of our results. And essentially here, on the left, you see Concerto. This is the test set from CPDB. And the right 2 scatter plots are just the first and second parts of the Concerto model. So if you provide just the fingerprint vector or just the Grover MLP, and then we find that, combined with both, Concerto does a lot better. And we also find that Grover by itself is actually quite variable. And it seems like there's a subset of molecules that it doesn't learn a good representation for. And these are some previous models that we compare against. And then, so this is a different database, so CCRIS. So this is collected in a slightly different way. And it unfortunately has only discrete labels. But here we also find that the full Concerto model does better than the individual components as well as the previous tools. And we were sort of interested in this role of mutagenicity pre-training, especially the multi-round pre-training. So we took the individual components of our model and individually added complexities such as adding just Grover, adding mutagenicity pre-training, or adding multi-round mutagenicity pre-training, this iterative task. And we find that through this whole process, the performance improves as we add these components. And then finally, this is sort of the part that I'm most excited about. And here we really wanted to see whether our model learns salient features. So to do this, we have this counterfactual approach where to try and understand if the model is learning relevant features, we find samples that are close in the input space, but far away in the prediction space to a sample of interest. And this essentially, to generate these related samples, it's a random walk in the string space representation of a molecule. So to sort of explain what's going on here, we need a distance function for measuring distance between molecules. So for that, we use Tanimoto scores. And as an example here, we have our original molecule here in the first row. And then using our trained model, we can get the prediction percentile in terms of carcinogenicity. And then the distance from the model to itself would be 0. And then we can use standard string mutation algorithms to change the nature of this molecule and generate thousands of molecules close to our original one, and use the model to evaluate the prediction percentile of the resulting molecule, and then get the Tanimoto score distance away from the original molecule. And then we can sort of visualize this in this plot where here in black, we have the original molecule, and then we're plotting the distribution of the other molecules that we're generating with this random walk. And on the x-axis, we have the Tanimoto score distance, and on the y-axis, we have the carcinogenicity percentile that predicted with our model compared to the test set. And here in blue and red, we identified 2 specific molecules that we're interested in. So molecules that are over a certain threshold, as indicated by these faint red lines, that are different enough in their prediction, so far away in the output space, but are close to the original molecule on the x-axis, so close in the input space. So down here, these molecules are actually visualized. So this is the base original molecule. And for the blue one, we're calling it the negative counterfactual. So a molecule that's close, but the model thinks is a lot less carcinogenic. And for example, you can see that the removal of this iodine group and a replacement with a hydroxyl group, the model would think it would make it less carcinogenic. And for the positive counterfactual, we can add this nitroso group, and then the model thinks that would make it more carcinogenic. And we can sort of expand this analysis to the whole data set wide, where there are these molecular substructures called toxicophores that people have previously noticed are associated with more carcinogenic molecules. And for every molecule in our test set, we can generate a positive counterfactual pair and a negative counterfactual pair. And then what we can do is count the percentage of the time the substructure appears in the original molecule, the percentage of the time it appears in the positive counterfactual, and the percentage of the time it appears in the negative counterfactual. And we see that almost across all of these toxicophores, if we ask the model to return a similar molecule to the test set, but either, for example, less carcinogenic as in the negative counterfactual case, or more carcinogenic in positive counterfactual case, the percentage of the time the carcinogenic subgroup is included in this new molecular structure increases for the positive counterfactual. So this percentage of the time that the subgroup is present in our molecules for more carcinogenic, the model thinks are more carcinogenic than the test set, this percentage is higher than the original. So intuitively, we can sort of say, if we ask the model to return a more carcinogenic molecule that it thinks is more carcinogenic, but is similar in structure, the percentage of the time this toxicophore is present higher in the positive counterfactuals and lower in the negative counterfactuals, which is sort of an orthogonal way to validate that our model is learning salient features. And overall, we think that molecular carcinogenicity prediction is an important problem, and it'd be great to get this right. And it's important to think about compound dosage while assessing model performance. And currently, our performance doesn't achieve levels needed for clinical decision, but we hope that this pre-screening approach can be used for selecting molecules for downstream carcinogenicity experiments. And then finally, in data constraint tasks that we so often have in biology, we find that large models trained with self-supervised learning using those embeddings can be useful for these tasks. Pre-training unrelated, but even sometimes lower quality experiments can be helpful. And encoding domain expert knowledge, in our case, in the form of molecular fingerprints, is also quite helpful. But still, the most effective way to improve performance is to acquire more data. And I think on this front, for carcinogenicity, there are reasons to be optimistic. So CPDB last was updated in 2007, and the mutagenicity data set is from 2009. So if someone has access to proprietary data or other data that they wish to share or retrain our model on, we'd be very happy to help. So yeah, thank you very much, and I'll take any questions.
Thank you, Philip. We have time for 2 or 3 questions, so go ahead.
Okay, there is one from Fradkin. What's the benefit of computing distance for the counterfactual analysis in a different space from your model inputs?
Right, yeah. So, I mean, the point of this analysis is to find a molecule that's similar, but the model thinks is carcinogenic or non-carcinogenic. So, I mean, we know that there are molecules out there that are like either very carcinogenic or not carcinogenic at all. So we want to really constrain the space. So like in a local neighborhood, distance neighborhood around the molecule, we'd like to find one that increases the carcinogenicity a lot or decreases carcinogenicity a lot. So it's like it's a way to constrain the space.
I'll just bend over. So the computational modeling is very impressive. I want to throw a bit of a curveball question. There's been some discussion in the chemical informatics world about ethical and real-world considerations about models like this. I was wondering if you thought about any practical ethical questions or real-world risks of a model that could accurately predict carcinogenicity of novel compounds, and if somebody could take a model like this and use it to score the billions of commercially accessible molecules from a vendor like ISMB or something, and try to find some super carcinogen with very high effect size. Is that something we should be thinking about?
Yeah, I think that's a very important point. As I guess these models of predicting real world, some sort of phenotypes get better and better, in theory, it's going to be individuals are going to be empowered to identify samples that are like really effective at whatever phenotype that you're modeling, and it can be used for good and for bad. Yeah, I don't have a great answer to like how we can prevent it or regulate it, but you know, the hope is that these things can be used for good. Yeah.